That second thing is the molecule needs to be rigid, okay? I'll talk about that a little more. And kind of the more rigid, the better. Right? So what we have in fluorescence is that we have this concept called quantum yield, which sounds fancy, but what it means is the quantum yield is essentially the ratio of the excited molecules that fluoresce. All right, right. So one is about molecules excited, and two is about molecules that actually fluoresce. And so we have that because it's quantum, of course, we have fancy Greek letters for it. And so quantum yield is represented by one of these. What is that? Phi? Great. Actually, we see it for phi psi angle, so I should know that, right? But so we got a phi. Here we're using phi for this. And so, for example, this isn't the example of the molecules that we're using in lab, but this is a great example. This molecule is called fluorine. And this guy has a quantum yield of 1. It's very rigid. So every molecule of this that gets hit, that is excited, will actually fluoresce. If we compare that to bipyridine, bipyridine looks like this. We get a quantum yield of 0.2. So only 20% of the molecules, because this can rotate around, only 20% of the molecules that are excited here actually fluoresce. All right? And so for your experiment, you only had one molecule that could actually absorb that light, right? It was the NADH. But if other things had absorbed that light, you know, here we only have one bond we can rotate around. But you could imagine if instead you were looking at lactate. Actually, that's more rigid than I would have thought, huh? Right? This, because of our pi structure here, we're going to be rigid through here, but we can spin around there. Well, I guess lactate probably, maybe it isn't a good example for rigid. Let's go ahead and look at pyruvate, though. I'm sorry, that was pyruvate. This is lactate. I mean, regardless, I can actually spin this methyl group, right? And I can get lose some energy there, and I can spin around here. We're still actually going to have this be nice and planar. But we still have more flexibility, certainly, than we have in this bipyridine, where we only have one bond we can rotate around. We can rotate here, we can rotate here. So here we only have the one. Questions? Okay. So those are those two qualities you need for something to fluoresce. Now, if you should hopefully have your worksheet. I did not bring extra. You'll have to get out a piece of scratch paper and work on that if you don't have that. We're going to go ahead and work through this worksheet I handed out yesterday, which essentially gave us a bunch of kinetic data, and we're going to plot it, and we're going to determine our Km and our Kcat out of that. Now, I'd ask you the significance of those numbers, but it's actually part of the worksheet, so we'll do that when we get to it. So yesterday we talked about, to do this, we're going to have to create a Lineweaver-Burke plot, right, if we want to get our data out of this. Okay, this is this reaction we've talked about before. It's how creatine can be used to regenerate ATP. This is really important. I actually went back and looked at the creatine literature, and it was kind of hit and miss. So I don't know what the answer is, whether creatine works or not. I thought I'd convinced that I had it, and I was talking to Dr. Sursky on a walk, and she said, "Yeah, how many people are in each of those studies?" She knows how to read the scientific literature for statistical biases better than I do, and she talked me out of taking creatine. So, anyway, There aren't really any side effects though. So anyway, so what we need to do here though is we want to do a Lineweaver-Burke plot. Remember, a Lineweaver-Burke plot is 1 over substrate concentration versus 1 over our initial rate. These are not 1 overs, right? So the first thing we need to do is we need to do 1 over for all of these. All right, so why don't you go ahead and do that? I'll do it with you. Actually, let's have you guys do the 1 over for the right-hand side. I'll do the left-hand side, and then I'll just have you give me numbers. Okay, anybody have some numbers for me here? 1 over 1083? 0.0923. Okay, next. 0.0539. Next, somebody else. Just for full participation. 0.0374. Next. 256. And then the last one? 0.020. Okay, great. All right, so the next thing we need to do, so this right here then would be 1 over V naught. Notice I didn't care about turning this into molar. I'm just going to carry my units along, all right? So we care what the units are, but we actually don't need to change them to molar, and we don't need to actually make them match. We can still get all our data out of this. So it's nice from that perspective. Now you need to make a graph, right? So you're going to make a graph where you put 1 over ADP on the x-axis, and you put 1 over V naught on the y-axis. And like I said, for your final, you will need to do this by hand. So let's go ahead and practice it now. Do you guys still have your graph paper? Even if you don't have your graph paper, you'll get pretty good answers if you make graph paper. So that's what I'm going to do on the board. I'm going to go ahead and I'm going to take a picture of this. You guys have all these numbers? I take a picture of this, and I'm going to find myself some white space. And I'm going to show you the power of graphing where you make your own graph paper. Does anybody need paper and a pen so you can practice if you forgot to bring your other stuff? I really recommend. This is great practice time. Okay, so in terms of looking at this, you want to, when you make your graph here, you want to try to use as much of the space as possible to get the best results. Okay, so you see what I've tried to do here? I've tried to spread things out nicely. And then, of course, if I do one of these points, it's trying to text somebody. All right, so if I do one of these points, we've got our first point is 0.05 and 0.923, right? So 0.05 up to 0.923. When's the last time somebody made you graph by hand? Years. So this is just awesome. Anyway, you might be out of practice. All right, how are y'all doing? You want some more time? I'll give you a couple more minutes and then we're going to go on and try and solve some stuff with it. Okay, why don't we go ahead and take a look at this? So easiest thing to pick off of this, let's see what I asked next. So calculate the Vmax. That's the easiest thing to pick off of this. So if you remember from your Lineweaver-Burke equation, it's 1 over V naught is equal to Km over Vmax, 1 over our substrate concentration plus 1 over Vmax. All right, and so this is a straight line equation. Y is equal to mx plus b. And so what is Vmax? Where is that on this graph? It's the y-intercept, 1 over Vmax, right? This right here is 1 over Vmax. And so looking at my graph, it looks like it's about 0.02. Might be slightly different on yours, right? And to put units with it, oh, I forgot my units over here. The original units. Millimole per second, right? So the units over here on this graph are going to be seconds over millimolar. Okay, so it's in concentration. That's a big M, second one. Okay. All right, so 1 over Vmax is equal to 0.02 seconds over millimolar, right? So I'm just going to take the inverse of that. So that's going to be 50. Is that 50? So Vmax equals 50 millimolar per second. All right, so that could be in moles per second or whatever. A millimolar, I have to change that to moles per second. If I was going to go and try and turn that into activities, like for your lab report. But it's fine here. Whatever volume we've got with that much enzyme, we're getting 50 millimolar of product per second. Okay, does that make sense? What we've done there? So that's our answer up here. Unless up here in the problem it asked for it in specific units, and it didn't. Now I want to calculate a Km for your Lineweaver-Burke plot. Make sure you have the correct units. All right, you notice that I took my graph and I extended it. I didn't tell you about this ahead of time, so it may not be easy to do on your graph. But you notice I extended it out until it hit the x-axis. Let me show you why. Okay, we can essentially solve for Km by using the x-intercept. So 1 over V naught is equal to Km over Vmax, right, times 1 over substrate plus 1 over Vmax. So what I want to do here is I'm going to, at the x-intercept, what's the value of 1 over V naught? Let's go back and look at our graph. So when I'm here, what's the value of 1 over V naught? 0, right? So what I'm going to do is for the x-intercept, this right here is 0. I haven't done this in a while, so I didn't practice it beforehand, so I'm hoping that algebra won't let me down. But we need to solve. We want to solve for Km, right? Okay. So what we're going to do is we need to get Km by itself. So to do that, I'm going to. What am I going to do? I'm going to subtract 1 over Vmax from both sides. All right. Let's go ahead and subtract 1 over Vmax. 1 over Vmax. All right, and so if I rewrite this thing, then I end up with 1 over Vmax negative is equal to Km times Vmax 1 over substrate concentration. Okay? All right, I'm still trying to solve for Km. What can I do here? I'm feeling pretty happy because it looks pretty good. All right. Multiply by Vmax. If I multiply both sides by Vmax, since I have Vmax on the bottom on both sides, right? Both Vmax's are going to go away. All right, and I've got -1 over here is equal to Km times 1 over substrate concentration. Now what can I do to isolate Km? Multiply by substrate concentration. Okay, and so now I get negative substrate concentration is equal to Km. Okay, but what am I plotting? 1 over substrate concentration, right? So I need to have, if I'm going to figure out how Km's related, and I'm plotting 1 over substrate concentration, then what I need to do is I'm going to have to I'll just divide both these into 1. So 1 over substrate concentration is equal to 1 over Km. And then I can flip this negative sign. So, because I'm reading 1 over substrate concentration off that x-axis, so that's equal to -1 over Km. You guys got that from the algebra? So what this is if I wanted to solve Km from the slope, right, that's equal to